Hello, students. Welcome back. Now, in our previous explainer, we tackled the basics, what reproduction actually is, why it's so absolutely crucial for survival, and the big differences between asexual and sexual reproduction. Today, we're zooming in on asexual reproduction. Think about it for a second. Have you ever wondered how bacteria can multiply into literally millions within just a few hours? Or how a tiny bit of yeast makes a massive lump of bread dough rise? What about taking a simple stem cutting and growing a brand new rose? Or how a broken piece of a hydra can develop into a totally complete organism? Well, it turns out all these everyday biological mysteries happen through asexual reproduction. Let's understand every single method one by one. So, welcome to our solo survival guide. Today, we're breaking down the fascinating mechanics of asexual reproduction. We're going to explore the whole shebang, from microscopic single-celled life forms to the macroscopic, beautiful plants in our gardens, figuring out exactly how they clone themselves to keep their species alive. Here is our game plan for today. First, the blueprint. Then, micromultipliers, budding and breaking, regeneration, spore survival, and finally, vegetative propagation. All right, let's jump right into Section 1, the blueprint, understanding asexual basics. Okay, so what exactly is asexual reproduction? The crucial point is right here in the definition. It's a process that relies on just one, single parent organism. And notice that last part, without the formation or fusion of gametes. Since there is zero mixing of genetic material from a male and a female, the offspring produced are pretty much identical to the parent. In biological terms, they are clones. Now, what's really fascinating here is how these specific traits act as a lightning-fast survival hack. It's super common in bacteria, fungi, algae, and certain plants. Think about it. Only one parent is involved, no fertilization occurs, no male and female gametes are formed. You just bypass all those complex steps of sexual reproduction. The result? A super fast process that pumps out genetically identical clones at incredible speeds when conditions are right. Moving on to Section 2, micromultipliers, the fission process. So, binary fission. This is literally the simplest method of reproduction out there. It's used by microorganisms like amoeba, paramecium, and bacteria to multiply very quickly under favorable conditions. It's an easy four-step process. The parent cell simply grows, right? Then, its nucleus divides directly into two. After that, the surrounding cytoplasm splits right down the middle. Boom, you're left with two completely equal, identical daughter cells. Now, to clear up a really common student doubt, let's look at this stark difference between binary and multiple fission. Binary fission, like in our amoeba example, yields exactly two daughter cells. Pretty straightforward. But multiple fission, that's a whole different ballgame. Here, the parent's nucleus divides repeatedly first, and cytoplasm surrounds each one, meaning one parent cell divides into many daughter cells, all at the exact same time. Plasmodium, the parasite that causes malaria, uses this trick to rapidly multiply. It's an extreme multiplier effect. Next up, Section 3, budding and breaking. Scaling up complexity. This right here is budding. It's the exact same process baker's yeast uses, which, by the way, is how we bake bread and make bakery products. It's also how the freshwater hydra reproduces. Basically, a small outgrowth, literally a bud, appears on the parent's body. It grows, develops into a complete miniature organism, and then it just pinches off. It separates from the parent to go live its own life. Meanwhile, other simple organisms like Spirogyra, which is a type of green algae, use a method called fragmentation. And it's exactly what it sounds like. The parent organism simply breaks apart into fragments or pieces. There's no budding involved at all. The body just snaps, and each individual fragment then grows into a whole new complete organism. All right, Section 4, regeneration, nature's sci-fi trick. Okay, listen up, because this is a massive exam topic. You have got to know the difference between fragmentation and regeneration. So, fragmentation is a natural biological process. It is the organism's intended way to reproduce, like in Spirogyra. Regeneration, on the other hand, is the ability to develop a complete body from a broken body part. It only occurs when simple organisms, like planaria or hydra, accidentally get cut or broken. The broken part actually regrows into a new organism. Just remember, regeneration is not possible in all organisms, only those with simple body organization. It's more of an accidental survival mechanism than a routine reproductive cycle. Moving to Section 5, Spore Survival, the survival capsules. Look at how bread mold, specifically an organism called Rhizopus, reproduces. It uses spore formation. Spores are these tiny reproductive structures that develop safely inside a protective casing called a sporangium. When that casing bursts, the spores are released. And they have some serious advantages. They are incredibly light, so they're easily dispersed by the wind. They're also tough enough to survive really unfavorable, harsh conditions. Because they're produced in massive numbers, they just wait around until conditions are favorable to finally germinate and grow into new organisms. Finally, Section 6, Vegetative Propagation, the botanist's toolkit. Now we are talking about plants. Vegetative propagation is when new plants develop from vegetative parts, like roots, stems, or leaves. Let's compare the natural methods with the artificial ones. Naturally, a potato or ginger uses its stem. An onion uses its bulb. Sweet potatoes use roots, and the bryophyllum plant incredibly sprouts new plants right off its leaves. Artificially, we use cutting for roses and sugarcane, just planting a stem in the soil. We use layering for jasmine and bougainvillea, bending a branch into the dirt until roots form. There's grafting for mango and citrus plants, where you join stems together. And then there's tissue culture, growing tiny pieces of plant tissue in a nutrient medium under sterile conditions, which is amazing for producing tons of disease-free plants. So what's the big takeaway here? Well, let's look at the trade-offs of vegetative propagation. The pros are fantastic. It produces identical plants, which perfectly maintains desirable characteristics. The plants grow much faster than from seed, you can produce large numbers of them, and it's super useful for seedless plants. But check out the cons. Because there is absolutely no genetic variation, every single plant is a clone. This means if a new disease hits, it spreads easily and could potentially wipe out an entire crop in a matter of days. All right, almost done. Let's do a quick exam revision. Board exam checkpoint. Take a quick second to mentally check off these key topics, as they are absolutely critical takeaways from our explainer today. You need to confidently differentiate binary versus multiple fission. You need to contrast fragmentation versus regeneration. Make sure you can define asexual reproduction and its characteristics, explain spore formation in Rhizopus, and identify those artificial vegetative propagation methods we just talked about. If you've got those down, you are in great shape for your exams. Before we wrap up, I want to leave you with this provocative thought. If a species relies entirely on asexual reproduction to create millions of perfect clones, what happens to that whole species if its environment suddenly changes? It really highlights the hidden dangers of having absolutely no genetic variation, doesn't it? And actually, that perfectly sets the stage for exploring why more complex life ultimately evolved to require two parents. Thank you so much for joining me on this deep dive. Keep questioning the world around you, and I will catch you in the next explainer.